Okay, good morning. Welcome back. We are working on either assignment 8 or the differentiable assignment number 3. Any questions about the assignments, differentiable or otherwise? All good? Okay, so we are working our way through challenges in the field, and we are dealing with one challenge which has to do with encodings, and by encodings I mean genetic encodings. How do we go about describing the thing that we're trying to evolve in a data structure to make it easier for evolution over evolutionary time to gradually improve those things to do whatever we want them to do? That can be robots, that can be neural networks. It's very challenging to get this right. And we looked at one of the problems of why this is so problematic. Last time we motivated our discussion by thinking about a population of evolving neural networks that are potential, unbeknownst to us as evolution is proceeding, these neural networks are struggling with the competing conventions problem. They're evolving to solve whatever we want these neural networks to solve, but within any one of these neural networks, they're solving different parts of the problem at different locations within themselves. This is true of us as well. None of us are perfect. Some of us are better at other things. And some of the reasons for that is because of your genetics. Hopefully, if you have offspring, you are able to bring together the best of you with your partner into your offspring. It's a notoriously difficult problem to get right. So we started last time by talking about the HyperNEAT algorithm. Neuroevolution, so we're not talking about evolving robots yet. We will at the end of this lecture. Neuroevolution of neural network topologies, the number and distribution of neurons and the number and distributions of synapses. And this evolutionary algorithm is going to augment these topologies. It's going to be able to add and remove neurons and add and remove synapses. So the topology, or the cognitive architecture, both synonyms for the same thing, are not fixed in HyperNEAT. And as you're going to see in a moment, HyperNEAT is designed to solve the competing conventions problem. Okay. HyperNEAT is a more complicated evolutionary algorithm than any we've seen so far. Instead of a simple vector, inside every genome are two different lists, and each element in this list itself is complicated. It's got many numbers associated with it. What are these two lists? Just to remind you, the first list describes all of the nodes or neurons that make up the neural network that's encoded in this genome. So in this case, this genome is encoding information about 5 neurons, 1, 2, 3, 4, 5. And the second list describes all of the synapses, 1, 2, 3, 4, 5, 6. But you'll notice there's only 1, 2, 3, 4, 5 synapses. In the description of each of these 6 synapses, there is a binary flag that indicates whether that synapse should be expressed in the phenotype, expressed in the neural network as well. You also, within your genome, have many, many genes that are never turned on. They were expressed in your ancestors, but they're turned off in you. So this is capturing a biological detail. None of what I've said so far has anything to do with the competing conventions problem. We'll get there in a moment. Okay, you'll notice that the description of each synapse is described by 5 numbers: the presynaptic neuron, where the synapse starts, and the postsynaptic neuron, where the synapse ends. So this particular synapse connects neuron 1 to neuron 4. Here's neuron 1, and there's a synapse that connects it to neuron 4. There's the synaptic weight. We've seen this before. This is usually what evolution is evolving. This synapse is turned on. And then there's this 5th number, which is called the innovation number. We'll come back to this in a moment. Okay, so here's a blueprint. Here's a genome. And then here's the phenotype. Here's the thing that this blueprint encodes. Okay, that's the encoding. Like all the other evolutionary algorithms that we've seen before, remember there's always a population of these things, and each one of them encodes one of these things. We're always dealing with a population. Okay, some other details of an evolutionary algorithm we always need to introduce is how does variation occur? When some of the individuals in the population are deleted, and the survivors produce randomly modified copies of themselves, how does that random copying occur? So in the NEAT algorithm, we're now going to look at how it does mutation. So let's assume that this particular genome produces this particular phenotype, and whatever it is that we want this neural network to do, it was doing a little bit better than other individuals in the population at that time. This is a survivor. From now on, I'm only going to show you the synapse list, not the node list. We're not going to look at this because all the interesting action in NEAT happens in the synapse list. Okay, this is just to simplify the figure. Okay, so we can see there's 1, 2, 3, 4, 5 encoded synapses. One of them is turned off. 1, 2, 3, 4. Okay, makes sense. All right, so there are many different mutation operators in NEAT. In all of the evolutionary algorithms we've seen so far, there's usually just one mutation operator, which is choose a gene and change that synaptic weight by a small amount. In NEAT, because it's a more complicated algorithm, there's different ways in which the genetic information can be randomly modified. Here's one of the many mutation operators, which is to add a synapse or add a connection. When this particular list is copied, this particular mutation event occurs at random and tacks on a new synapse. And this new synapse randomly connects neuron 3 to neuron 4. Okay, so we can add a connection. Here there's a mutation event that between the synapse that connects neuron 3 to neuron 5, there's a mutation event that can also add neurons. So we've seen add synapse, add neuron. As you can probably guess, there's two other mutation operators, randomly delete or disable a synapse, randomly delete or disable a neuron. That's how the augmenting topology happens. The topology is not fixed. Evolution itself can randomly add and remove neurons and add and remove synapses. So now I want you to mentally imagine a population of neural networks that have different topologies, have different cognitive architectures, and over evolutionary time, those architectures are going to change. Some architectures are going to survive and some aren't. Remember our discussion about neural networks when we talked about exclusive OR? You needed two hidden nodes inside in order to compute exclusive OR. If we had evolved neural networks and we wanted them to compute exclusive OR, and we hadn't built in any hidden neurons, without augmenting topology, we're in trouble. No matter how long we run the evolutionary algorithm, it's never going to evolve a neural network that computes exclusive OR. Here, hopefully, through random mutation events, eventually NEAT will stumble across a neural network with two hidden neurons inside, and hopefully with enough evolutionary effort, those two hidden neurons and the synapses flowing into them and out of them would gradually become better and better able at computing exclusive OR. We drop these neural networks inside a robot to control a robot to do bipedal locomotion or object manipulation. We don't know what the appropriate topology is. How many neurons and synapses are required to do this or to do this? Unknown. We want the evolutionary algorithm to figure it out. So far, so good? Questions? Okay, these are mutation operators. There's other familiar ones like randomly choose a synapse, find the weight inside, and then randomly modify the weight. Several other mutation operators, not really important for our purposes. Okay. Let's talk about the innovation number now. When we add something new, like add a new synapse or add a new node, inside the NEAT algorithm, we're going to keep a global counter. So inside the code base, there's a global counter that starts with 0, and every time, the first time a new synapse is added to a neural network, anywhere in the population, that 0 is assigned as the innovation number to that synapse, and the global counter is incremented by 1. The next time a mutation operator adds a synapse, a 1 is assigned as the innovation number for that, and so on and so forth. So every time a new thing is created, it gets a unique identification number. This is true for neurons and synapses. They're not showing this innovation number or this historical marker in this cartoon, but every time a new piece of genome is added, we assign this number. Why? Okay. Let's go back to the example of this mutation event that occurred at some point during the evolution of NEAT. We've added synapse 7 here that connects neuron 3 to 4. I'm sorry, I misspoke. The integers at the top here, these are the innovation numbers. So at this moment in time when this synapse was added, this global counter in the code base was currently at 7. That was the ID number that had yet to be assigned. So it assigns an ID number or a historical marking of 7 to that synapse. Let's imagine that for whatever reason, this particular synapse by chance happens to increase the fitness of this neural network compared to its parent. Again, just by chance. It was a little bit better. So this individual survives as evolution continues, and let's imagine this less fit parent dies off. Even later in evolution, imagine that this individual produces 2 offspring, and that offspring inherits these synapses. So now we have 3 individuals in the population, the parent and the 2 children. All of them have this red synapse inside them. They have a synapse that connects neuron 3 to 4. And that synapse in all 3 of them has the same identification number, 7. As evolution proceeds, those 2 children produce children of their own, grandchildren, great-grandchildren, great-great-great-grandchildren. And in that lineage, other events start to happen. Neurons are added and removed, things are moved around. So eventually, if you start to look at the collection of great-great-grandchildren, you might see a synapse with a historical marking of 7, but that individual synapse may appear in different places inside all of those relatives at different positions. Later on, if those individuals were to come together as 2 parents and we want to recombine that genetic information, we don't want 2 copies of synapse 7 to end up in the offspring of those 2 parents. We also don't want synapse 7 to not appear at all in the offspring of those 2 parents. Synapse 7, for whatever reason, kind of helped the original ancestor back here and all of its offspring. So presumably we want the offspring to keep inheriting just 1 copy of synapse 7. So the reason for this historical marking, and this is what makes NEAT neat, is no matter where these things occur in the neural network, when we come and we cut 2 neural networks in half and glue the 2 halves together, we increase the chances that that offspring inherits 1 and only 1 copy of useful stuff. Everybody see the intuition behind that? Okay. Let's see this in action. So we just talked about mutation. So now let's look at crossover in NEAT. This is what NEAT was really designed to do. Let's imagine at some given point in evolutionary time in the population we have this neural network and this neural network, these 2 survivors, and they're randomly chosen for a crossover event. We grab them and we're going to try and cut them apart and glue their parts together to produce 1 offspring that hopefully inherits the best parts of the 2 parents and leaves the worst parts behind. Okay. Remember that these 2 neural networks are encoded in the genome, and again, we're only showing the part of the genome that encodes the synapses. So parent 1 has 4 enabled synapses, 1, 2, 3, 4, and parent 2 has 1, 2, 3, 4, 5 enabled synapses, 1, 2, 3, 4, 5. These are just sanity checks. Okay. Obviously, just looking at these two neural networks, they have different topologies. They have different cognitive architectures. They have different numbers of neurons and synapses, and they're arranged differently. We've got a Mac over here and a PC over here. They're different, so this is going to be tricky. We've got to try and cut them carefully. How do we do this? We're going to take parent 1, and we're going to stretch parent 1 over here in the following way. We're going to start with the leftmost element in parent 1's synapse list. This is synapse number 1. It has an innovation number of 1. We place it here, and then we go to parent 2, and we look within parent 2 to see if it has a synapse that has an innovation number of 1, and it does. It also has a copy. This means the fact that these two parents have a synapse with the same historical marker means what? What does that tell you about the ancestor of these two parents? I gave you half the answer already. They're related, hopefully distantly related. They share a common ancestor, and that ancestor had one and only one copy of synapse 1. That synapse also exists in both of these hopefully distant cousins. It's probably a good thing to have. All three of them had one copy of it. So we don't want a child to have 2 copies of this synapse, and we also don't want this child to have 0 copies of synapse 1. So we take parent 2 down here, and we place synapse 1 underneath synapse 1 from parent 1. We're kind of lining up horizontally things that existed in the common ancestor and therefore probably play similar roles in these two parents. Maybe not identical, because other evolutionary things have happened, but these are probably similar parts. So I've got a Mac up here. I'm going to grab your PC and I'm going to get out my chainsaw and start cutting. I'm going to try and cut carefully. I'm going to find the CPU in my Mac and cut out the CPU. I'm going to find the CPU in your PC, and I'm going to cut out your CPU. I now have a Mac CPU and a PC CPU. The dumbest thing I could do is put both CPUs into the new computer I'm trying to build. The other dumb thing I could do is not put a CPU at all in this new computer. I've got 2 things that I think are performing similar function, and I'm going to flip a coin. I'm going to put one of these 2 things into the child. So in this case, NEAT also flips a coin. Heads, it puts parent 1's copy of synapse 1 into the offspring. Tails, it puts parent 2's copy of synapse 1 into the offspring. Did it flip heads or tails in this case? Checking to make sure you're all still following me. Yep. Tails, right? So tails came up. We're going to put parent 2's copy in here. Okay, done. We've copied one gene from the 2 parents into the offspring. We go back to parent 1. We hop 1 step to the right. Now we have synapse 2. I'm going to just call them now synapse 3, 4, 5, where the number references the innovation number or this historical marking. So synapse 2, we place it next here. We go looking for synapse 2 in parent 2. There's a copy. We put it underneath. Flip a coin again. In this case, it's heads. So we put parent 1's copy of synapse 2 into the offspring. And we keep going. And it turns out that the next one, synapse 3, has no corresponding copy in parent 1. So we stretch parent 1's genome. We leave a gap here. And now we don't flip a coin because there's only one copy. So we just take that copy and drop it into the offspring. Synapse 4 exists in both parents and it's disabled in both parents. What is sort of the hint that NEAT is giving us here? Synapse 4 existed in the common ancestor. Was it on or off in the ancestor? There's parts of your genetics that are turned off that are also turned off in me and everyone else in here. Why?
They helped at one point, and then to do what's currently trying to do, it doesn't actually need it anymore.
It probably was useful at some point and got turned off. So whether the offspring actually needs it or not, who knows? But we keep following the neat algorithm, the logic. There's 2 copies of it. We line it up, flip a coin, heads. We bring in one of these 2 parents. Copy. It's not even clear from this picture which one it is. If it was useful in the past, could be useful again in the future. Who knows? Okay, so we keep going, and we're now going to refer to these genes as disjoint genes. This gene here for this synapse is disjoint. It doesn't have a copy in the parent. And then there are also excess genes. So 7 and 8 over here have innovation numbers that are higher, 7 and 8, are higher than any innovation numbers in parent 1. What does that mean? What do you think about these particular synapses that only appear in parent 2? This one's a little trickier.
They help achieve something that specifically parent 2 is good at, but parent 1 probably doesn't do.
So between whatever the common ancestor is of these distant cousins, if we follow the lineage from that common ancestor down to parent 2, somewhere along that evolutionary path, 7 and 8 got added, and they're still there in parent 2, so they're probably good for parent 2, however parent 2 is trying to solve the problem we've set for these neural networks. Who knows? So INNEAT always tries to err on the side of caution. We just toss them into the offspring. They were useful for one of the parents. We toss them in. So at the end of this example, we have an offspring with 1, 2, 3, 4, 5, 6, 7, 8 synapses. 2 of them are disabled. 1, 2, 3, 4, 5, 6. All right, that makes sense. So we now have an offspring which has a different topology, has a different cognitive architecture compared to the 2 parents, but we've ensured that it's received one and only one copy of things that probably do similar things in each of the 2 parents. That's the intuition underneath the NEAT algorithm. Okay, question?
Yeah, if they've been given, if we've evolved the same synapse, but they were given different identifiers, we only include one of them.
Only include one of them. That's right. So this is not a perfect process, right? This is just trying to increase the probability that if I cut my Mac and your PC in half and put them together carefully, I increase the chances that the offspring will be as good as or better than the parents. As you can imagine, still, most of the time, the child is not as good as the parents. Crossover is always a very risky enterprise.
Does it have to be 50-50 when you pick? I just imagine you could have like some sort of heuristics for like, this synapse is 0.1, this synapse is 0.8. Maybe we want to take the 0.8.
Absolutely. So this algorithm is 25 years old. There's been many improvements on it so far, but amazingly, it's still used in a lot of branches of robotics and AI still to this day. It works. It's not perfect, and there are many improvements you can think of, and that's a good one. Okay. Okay, we just talked about this. Okay, this is the frustrating point of lecture 13, is I'm not going to show you any results from NEAT. We actually need to bring this back to robotics. So you'll just have to trust me that this algorithm works very well. You can evolve neural networks that have different architectures, and it tends to work. What I want to focus on in the rest of lecture 13 is not NEAT, but HyperNEAT. As the name implies, this is yet another evolutionary algorithm that builds on top of NEAT. It's got NEAT inside. Yeah? Okay, so what is, and we're going to see how HyperNEAT is used to evolve robots and why HyperNEAT is really good for evolving robots. Okay, we're good with NEAT? Okay, here we go. So HyperNEAT, in a nutshell, is going to evolve neural networks. Here's one of them over here, but this is a special kind of neural network that we haven't seen before. These are known as compositional pattern producing networks. So it's a bit of a mouthful. We're going to go through what CPPNs are, but basically HyperNEAT evolves populations of CPPNs. Okay, so CPPNs, compositional pattern producing networks. What are so special about CPPNs? Like all the neural networks we've seen before, there are input neurons and there are output neurons. In the cartoon example I'm going to show you here, there's just 2 input neurons, 1 output neuron, and 1, 2, 3, 4, 5, 6 hidden neurons inside. Yeah? Okay, I'm going to draw for a little bit now. CPPNs are at heart a geometric neural network. They're dealing with geometry. So what does a CPPN do? A CPPN paints patterns across space. That's the pattern producing part of the name. Okay. You'll notice that the input neurons are called X and Y. So let's make a CPPN over here with 2 input neurons. And I'm going to make an even simpler CPPN than this one. We're going to have no hidden neurons, and we're going to have just 1 output neuron. And I'm going to call this output neuron B, where the value arriving at B is going to dictate how much black I put on the board. In a CPPN, we tell the CPPN, this is something we as the designers have to do. We have to define a space for this CPPN. I'm going to tell this CPPN that the space is X, and X is going to range between 0 and 1, and Y is going to also range between 0 and 1. The CPPN is going to take over at this point. It's going to take a bunch of locations spaced regularly in space. And it's going to take each of these coordinates, let's start with 0, 0, and it's going to plug 0, 0 into the input, into the 2 inputs. So let me move the X and Y out of here. We're going to drop 0 and 0 into the 2 input neurons. What's the value that arrives at the output neuron? We've got 3 neurons, no synapses. The CPPN is telling us to put no 0 amount of black at position 0, 0. Let's now go to position 0.1. So X is now 0.1, Y is still 0. What does this CPPN tell us to put at position 0, 0.1, 0? Another goose egg, right? 0. What pattern does this CPPN paint? You're shaking your head, which means nothing. The null pattern. It paints all white across this space. Not a very interesting CPPN. Remember that in NEAT, we're evolving populations of neural networks. Let's assume for the sake of argument, for some reason, this parent survives and produces an offspring, and a random mutation event adds a synapse with weight 1 that connects neuron Y to neuron B. We've now made a change to the genome. We made a change to the CPPN. We now want to see whether it's produced a changed pattern. Let's plug 0, 0 in here. What value arrives down here? 0 times 1, no matter what the activation function in here, is 0. So it places 0 here. 0.1, 0. Still 0. This whole row down here, all white. Let's now visit x equals 0, y equals 0.1. What do we get down here? 0.1 if we assume no activation function. So let's assume no activation function. And I'm going to represent that by drawing a little picture inside the neuron where the horizontal axis inside the neuron represents the raw weighted sum that's arriving into this neuron, which at this moment in time is 0.1 times 1. So we look up 0.1 on the horizontal axis, and then the vertical axis is going to tell us what value to actually emit from this neuron. Let's assume we have a linear activation function. Whatever the raw weighted sum is that comes in, that's the value that goes out. So yes, let's assume if we put 0 and 0.1 in here, it tells us to paint 0.1 amount of black at this position. X equals, sorry, am I doing this right? Yes. Okay, let's actually go up. X equals 0, Y equals 0.2. X equals 0, Y equals 0.3.
3.
So as we move up in this space, we get a darker and darker amount until we get total amount of black at the top. What about the next row over? X equals 0.1. What does this row look like? Same thing, right? Because there's no connection here. So what pattern is produced? A gradual darkening gradient as we travel up. Everybody see that? Okay, so we have a neural network that is painting a pattern. So we've got the pattern producing part. Let's try one more example. Let's imagine that child CPPN in turn produces this grandchild by chance. What pattern does this CPPN paint? A quick way to figure this out is mentally to plug in the 4 corners. Plug in 0, 0 to this neural network. Plug in 1, 0, x equals 0, y equals 1. So plug in 0, 1 to this neural network. Plug in x equals 1, y equals 0. So plug in 1, 0 to this neural network. And then plug in 1, 1 to this neural network. What pattern does this neural network produce?
It's like getting darker diagonally like this.
Okay. We get a darkening gradient as we travel up and to the right. Okay, so these are very simple CPPNs. I've introduced this little visual metaphor here of putting the activation function inside the neuron. Let's go back to this CPPN over here. You can see that there are, I'm sorry, I misspoke before. There are 5 hidden neurons inside, and this is the output neuron. What do these different curves inside these 6 neurons represent? What are they? We know what this one is, the identity function. This is no activation function. This is a different activation function. This says, if we look along the horizontal axis, this is the raw weighted sum arriving at this neuron, and it tells us what to emit from this neuron. So in CPPNs, there's several things that are different between CPPNs and the neural networks you've seen before. The first one is that they're producing or painting patterns across space. The second one is that NEAT, which is evolving these populations of CPPNs, NEAT is going to augment their topology. It's going to add and remove neurons and synapses. NEAT can also modify or change the activation functions inside these neurons, which seems like an odd thing to do. Let's go back to our simpler example over here, and let's put the sine function inside. And let's leave these synapses the same. Now it's a little trickier. What is the pattern that this produces? I hope you had your caffeine this morning.
Like, along the, like, up and right diagonal, it'll be like dark, then light, then dark, then light.
Exactly. I don't know if I'm a good enough artist to try and draw this. Let me give it a try. It gets darker, then lighter again, darker, lighter again. What happens if a mutation increased the frequency of the sine wave? Now what happens?
It's going to get darker and lighter more often.
More exactly. Okay, that's why they evolve the activation functions inside. It gives NEAT, which is the algorithm that's evolving these things, the opportunity to fine-tune or change these patterns. We haven't said anything about a fitness function yet, right? There's nothing good or bad about any of these patterns. We're just showing that they do this. So we understand 3 of the 4 letters in the acronym for CPPN. Let's tackle compositional. What is going on here? If you think about this hidden neuron here, it's transforming these coordinates, x and y, in a certain way, and then those transformations are being composed into another transformation. So one way to think about this is these neurons are doing coordinate transforms, they're altering things, and then those alterations are being composed. This one is acting on this one, which in turn is acting on this one, and so on. That's the compositional producing part. If composition of coordinate transforms goes a little bit over your head, that's okay for our discussion today. Just a little more technical details about why. Okay, so hopefully at this point you're like, why the heck would someone want to add all of this complication? We now have a relatively complicated evolutionary algorithm that is trying to evolve relatively complicated neural networks. What is all this for? You might have noticed that in our little cartoon example here on the board, I was making random changes to the CPPN, but that wasn't producing random patterns. We had gradations. We had gradients across space, or we had uniform white. You can also make uniform black. One of the beautiful things about CPPNs and why they're still used to this day is NEAT, like all other evolutionary algorithms, makes randomly modified copies to things, but that produces non-random changes. We don't have white noise here. There isn't a whole bunch of black here and a little bit of gray here, black here, gray. It's not random. We get certain gradations, we get symmetries. You'll notice that some of these activation functions are symmetric about the y-axis. If we look in nature, you are not a random collection of parts. There are gradations across you. You have symmetry. The left side of your body looks more or less like the right side of your body. Among the insects, there are things that look like sine waves, things that get big, then small, big, small, big, small. You get repetitions across space. That's why some of the activation functions in CPPNs are trigonometric functions, things that repeat. And then finally, they put a little cartoon here. You are bilaterally symmetric, but not perfectly bilaterally symmetric. If we look inside, there are things that are placed at unique locations. I don't have time to go into the details of this, but there are CPPNs that can paint a particular pattern at a particular place. It's not all repetition and gradients throughout. So CPPNs, even if they're changed randomly, they produce non-random changes. Okay, so to try and show this, they've got a little cartoon insect here, which is mostly bilaterally symmetric, and Gaussian neurons with Gaussian activation functions help with that. And inside the same CPPN, another neuron that is applying a sinusoidal pattern to the y-coordinate produces light, dark, light, dark, light, dark, light along the vertical axis. It's composing coordinate transforms to produce something, in this case, not an insect, but a pattern, or pattern of an insect that has bilateral symmetry and repetition and unique patterns that appear in only one place, all in the same pattern. Different neurons are contributing with these different things. So far, so good? That's why CPPNs were invented, to allow evolution to modify neural networks, but the things that those neural networks produce are non-random. Okay, so now with HyperNEAT, we have a pretty complicated algorithm. We have an evolutionary algorithm that is evolving, it's evolving sets of lists. Those lists are transformed by NEAT into neural networks, and then those neural networks, in turn, produce a pattern. So far, so good? Okay, let's take a moment to build up our intuition for CPPNs. We're about to apply them to robotics, but we need to have a better intuition for how they work first. I'm lazy, I've only got one color at the front of the board today. Let's say we wanted to create a CPPN that produced patterns across this XY space, but not in black and white, in color. What change would you have to make to the CPPN so that now it paints in color?
You need 3 outputs.
Good start. What are you going to call these 3 outputs? Okay, I'm not going to make any change. Take this CPPN, 2 inputs, no hidden neurons, 3 outputs. What pattern does it produce?
The same thing as before, it's green.
Absolutely, right? So now it's only dropping green onto the board. Let's add a few synapses. Minus 0.3. What does this CPPN produce? Don't break your brain, I don't know, right? It gets complicated very quickly. Whatever it is, it's going to be some wavy pattern in color. There's going to be some red, some green, some blue. Yeah? Okay. If you're interested, Google pick breeder. There's a website, and behind that website is running HyperNEAT. And pick breeder will show you not the CPPNs. It's going to hide the CPPNs from you. It'll show you a 3x3. It'll show you 9. It'll show you 9 different patterns, and it'll ask you, which one do you like the best? Let's say I like this pattern the best. I click on this one. What do you think happens under the hood?
The network that made that pattern survives and the other ones get chopped.
Absolutely. So under the hood, there are 9 CPPNs, and I said I like the 3rd one the best. So the other 8 get killed off. And NEAT, which is running underneath everything, produces 8 randomly modified copies of the pattern that I like. And now maybe this one is even a little bit better. I like this one a little bit more, and I click on it, and I keep clicking, and I keep clicking, and now pick breeder has enslaved a human, and that human is the fitness function. There is no fitness function except whatever this human happens to like. It's subjective. Yeah? Okay. Works pretty well for a website because these are two-dimensional images. What would happen if we wanted a CPPN to paint a pattern in 3-dimensional space? So now we tell the CPPN X, Y, Z.
Third input.
Exactly, right? Now off we go. We start evolving populations of CPPNs, each of which produces a pattern in 3-dimensional space. All right, let's try one more. I want to make a CPPN that creates a movie for me, an animation.
Just need time.
I'm going to change the Z to a T, which is now going to be an integer. So T0, I'm going to plug in T0, and then I'm going to visit all of these coordinates in 2-dimensional space. It'll produce a pattern for me, save that frame of the video. Same CPPN. Now get it to paint a 2nd frame for me, a 3rd frame for me, and so on. So one of the cool things about CPPNs is it can paint patterns in differently dimensioned spaces. XY, XYZ, XYT, XYZT. From the CPPN's point of view, the dimensionality of the space that it's painting the pattern in doesn't matter. So far, so good. CPPNs have been around for a long time. As you can imagine, they're kind of fun to play with. You can use them to paint music. Any musicians here? Imagine a blank sheet of sheet music. How would you get a CPPN to paint notes, chords?
I would have the entire note scale as an output, and then input probably time, just time maybe?
The musical scale, or the bars here, we would not have them at the output, we'd have them at the input,
Okay.
right? So we'd ask the CPPN to march down the musical scale and then paint a whole note, a half note, a quarter note. So the output would dictate what note, if any, to place at that position on the musical scale. And then, like we did with the movie, we might have a time index, which now goes to the next moment in time in this piece of music, if it is going to be a piece of music, paint more notes, and so on and so forth. Yeah? You can imagine that artists and musicians have had a great time with CPPNs over the years. Yes?
Would you need like a hidden layer there for the different types of notes, and then the input being the like note on the scale though?
Great question. Give it a try. There's no right answer here. There's different ways to do it, right? We're figuring out how to encode the space of music, all possible songs, and then we're asking a CPPN to paint a particular song in that space. Okay. This is not a music class. This is not an art class. What I want you to take away from these examples is the dimensionality up here. This is going to be important in a moment. Okay, let's push on. Okay, so why is it called HyperNEAT? The hyper refers to the fact that it's quote unquote painting a regular pattern onto a hypercube, right? Something, a space of arbitrary dimension, or a dimension that we set. So the dimensionality of the hypercube is determined by the dimension of the input coordinates. All right. Okay, finally, we're getting towards the end of lecture 13. We are now going to use HyperNEAT to paint neural networks into a robot. As I mentioned before, it's very difficult to know what the appropriate cognitive architecture is for a robot. So we're going to use, these authors used HyperNEAT to paint brains into robots. Okay, here we go. Let me just jump ahead so you can see. We're going to be dealing with a quadruped. This quadruped, it's got four hip joints, 4 knee joints, some sensors. Hopefully very familiar to you at this point. They're going to take all the circles that you see here. These are neurons inside the robot. And you can see immediately that they're embedding these neurons in a 3-dimensional space. Okay. This sheet of neurons here, these are the sensors, the sensor neurons. The middle sheet here, these are the hidden neurons. And in the back here is this third sheet. These are where all the output neurons are. Let's start with the sensor neurons. So each row here corresponds to each of the 4 legs, leg 1, 2, 3, and 4. Inside the hip of leg 1 and the hip of leg 2, hip of leg 3, hip of leg 4, they're placing a sensor that's going to report the current value of the hip's rotation forward and back. This is not very good terminology. These are proprioceptive sensors, or angle sensors. We've come across them before. So as the quadruped is moving around in the simulation, the value of the angle of the hip is being plugged into these sensor neurons. Similarly, as the hip rotates inward and outward, We're going to take, there's another angle sensor sitting there. So again, we've got a 2 degree of freedom joint at the hip that can rotate the leg forward and back and in and out. We've got proprioceptive sensors in the knee as well, so we're at 12 sensor neurons so far. We've got our familiar touch sensors that are in the 4 feet. We have three additional neurons that report the orientation of the robot. Pitch, roll, and yaw of the robot's main body. And then finally a sine wave. A neuron here that outputs a sine wave. We've seen something like this before. What is this?
Keeping time.
It's keeping time. There's a particular name for these kinds of neurons. You've got them in your spinal cord. This is the central pattern generator. Yeah, okay. So we've got 5 times 4, there's 20 sensors. CPGs are not really sensors, but whatever. 20 or so sensors. And then in the back, it's a little hard to see, we've got 3 times 4, we've got 12 motors that control the 3 motorized joints, 1, 2, and 3 in each of the 4 legs. These are things that the authors know the robot needs. They built the body of the robot. They need these input and output neurons, but the rest is unknown. They are now going to use HyperNEAT to paint weights onto the synapses that connect the sensor neurons to the hidden neurons to the other hidden neurons, and the synapses that go onward from the hidden neurons to the output neurons. We now have a ridiculously complicated evolutionary algorithm. We've got NEAT, evolving populations of things. Those things are CPPNs. Each CPPN, which is a network, is in essence creating another neural network, the one that controls the robot. Everybody with me so far? Okay, seems complicated. So you should be hoping that there's a reason for all of this complication. One last thing I want to say about this, just so you can integrate this in your head. Remember that for all of these CPPNs, we're going to evolve them, so we also need a fitness function. We need to say how good or bad each CPPN is in the population. What do you think the fitness function is here? This is going to be a neural network that controls a robot.
How well the robot does?
How well the robot does, right? They're going to, as always, evolve robots to move as far to the right as possible. So eventually, each robot is going to be controlled by one of these. We're going to measure how far it travels in the simulation, assign that fitness value back to the CPPN, and NEAT will decide who survives, who's killed off, who produces mutated offspring, and who gets together to produce crossed over offspring. Okay, everybody put all those pieces together in your head? Okay, why do this? What do you think was the hope here for this experiment?
Some type of evolved control emerges out of the patterns that are produced.
That some evolved control of the robot evolves out of the pattern of what? We were painting black onto the board. We were painting color. We were painting musical notes. The pattern of weights, right? So it's a little difficult to see, but imagine a whole bunch of arrows pointing from this sheet to this sheet, a whole tangle of arrows pointing inside this second sheet, which is recurrent synapses and self-synapses, and then a whole forest of arrows pointing outward to these 12 neurons. Those synapses are embedded in this three-dimensional space. So you can imagine a regular gradation, a regular graduation of weights across this neural network. That's the hope, or that's most likely what's going to happen by using HyperNEAT for this robotics application. But why? Why do you think that's going to be useful? This ties back to our discussion about legged locomotion.
Locomotion
has patterns. The Ministry of Silly Walks also has patterns, but they're kind of complicated. The silliest walk of all is when every stride is different from every other stride. Again, try this at home. It looks very silly. It's exhausting. It's a terrible way to move. So the idea, the intuition behind this experiment is let's bias the evolutionary algorithm, which in this case is neat, bias it to a subset of sets of synaptic weights. HyperNEAT is going to try and is mostly going to avoid the vast majority of sets of synaptic weights, which would be a random collection of values. And it's going to instead evolutionarily search the much smaller space of regularly patterned weights. That's the idea. And we're going to see whether that actually helps. We wouldn't be talking about this experiment if it didn't actually help. It helps. Okay. All right. We got one last detail, which is we haven't actually talked about the CPPN. So let's build it. What do we need for our CPPNs to be able to paint weights in this space? We're painting patterns in three-dimensional space, so it's a strong hint.
Probably gonna have 3 inputs, 2 for which neuron it's painting for, and 1 for the layer it's painting from.
Okay, we could do that. So I'm going to call the first one pre for presynaptic neuron. The second one post for postsynaptic neuron. If we assume all these neurons have integer indices, we're going to be plugging in integers here. And then finally the layer. So this is also an integer. That's one way to do it. What do we need at the output?
You would need an output for every neuron in the next layer. Or
Maybe. What are we painting? Remember, the output neuron is where the pattern gets output. It's the thing. B for black, RGB for red, green, blue. What are we painting in this case? We're not painting colors anymore.
One output.
Just one output, and what are we going to call it? W for weight, right? So if we plug in the names of 2 neurons here and what layer they belong to, or what layer the synapse belongs to, the value that arrives here, let's say it's minus 0.3, that synapse gets painted with that weight. Question? Comment?
Would you not need more inputs than that?
Ah, why?
Because the layer before, you would need 2 coordinates for which neuron, and then for the post, you would need 2 coordinates for the next layer.
Possibly, right? These indices are not strictly, they're not strictly coordinates, right? They're not arranged in the 3-dimensional space. It'll work. You'll still get a CPPN that paints patterns, but it's unlikely to be a pattern that has a gradual pattern or a repeating pattern across this 3-dimensional space. So a good start, but let's try and restrict our input neurons to geometry, to coordinates, places in this space. So this is right. We got this part down. Remember back here when I started with CPPNs, I walked across this 2-dimensional space. Let's imagine that I'm going to walk across this 3-dimensional space. There's a synapse connecting every neuron in this layer to every neuron in this layer. In this layer, there's a synapse connecting every neuron to every other neuron, including itself. And there is also synapses from every neuron in the hidden layer to each of the 12 motor neurons in the output layer. So we've got a whole bunch of arrows, which are the synapses, embedded in 3-dimensional space. How should we ask the CPPN to walk across this 3-dimensional space and paint a pattern as it goes?
XYZ coordinates.
XYZ seems the most natural choice. Which XYZs? There's an infinite number of coordinates in this three-dimensional space. The XYZ coordinates of what?
I guess I'd go, maybe Z would be the layer, and then the X and Y would be the neuron.
We could put in here the layer again, and XY would be the neuron, but there's always two neurons associated with every synapse. Can we be a little bit more elegant?
We do something with like, just looking up there, you got your one side is like your motors and the other side is your sensors.
Yeah.
So seeing like one is the motor and the other is the sensor for it.
Maybe, but again, motor and sensor, they're not strictly the geometry here. Think about the things we're painting. What are we painting? The synapses. I'm fishing now. Simplest thing would be the X, Y, and Z of the synapse. That's the thing we're painting. When I did this, I said, what's the black that I should paint onto this little bit of space? So similarly, we're saying, what's the weight that I should paint onto this thing, this synapse? Remember that these synapses, they're in this three-dimensional space. Now they're aligned because they start at a presynaptic neuron and they end at a postsynaptic neuron. So, I don't know, take the midpoint of that line. So for example, imagine the synapse that connects this neuron to this neuron. There's the midpoint right there. Let's take that XYZ and paint the weight onto it. Simple. Everybody see that? That would definitely give us a regular gradation of weights across the synapses in this neural network. Again, going back to you, inside your central nervous system and peripheral nervous system, as you move a small distance inside your brain or your spinal cord, you don't suddenly see completely different neurons and completely different synapses with different properties. There's a gradation. There are waves of similarity within your nervous system. This solution we've come up does more or less the same thing. One last twist on this before we get to the results. This actually has a bit of a problem with it. It's very, very simple, but there's a problem. Can anybody see what it is? Ideas?
It seems like you're going to get a bunch of these subgraphs that could be isomorphic, and so like we just might be getting into these holes where we're redesigning the same thing over and over again.
There's some redundancy and some constraints here. We're actually, by using this as our encoding for the CPPNs, we're actually tying NEAT's hands. There's some things that NEAT will not be able to do, or HyperNEAT will not be able to do with these synaptic weights.
You can't, no, never mind. Is it gonna be like the actual neuron that's gonna be defined by a certain space, like you can't really expand it, or?
We've defined a space, right? It's all these synapses, and they've got these weights. I want you to think about this literally, geometrically, right? This three-dimensional space. We've got synapses in this space. What's the problem here?
You can't have residual connections, like going directly from the input layer to the output layer?
That's true. We could add some in, and then this thing will happily paint them as well, because a synapse traveling from here to here has a midpoint. We've restricted ourselves to 3 dimensions. Good place to start. We're kind of comfortable. But what's the problem with that from HyperNEAT's perspective? Think about a synapse that goes from here to here, and think of a second synapse that goes from here to here.
It might share the same endpoint.
Not the same endpoint.
Right.
What do those two synapses share?
Same endpoint.
Nope, they've got different inputs and different outputs. Here to here and here to here. Two different synapses. I'm fishing.
They cross
Yeah, so,
the midpoint.
thank you. They have the same midpoint. So what's going to happen? When this CPPN paints these two synapses,
It's gonna paint the same way for both synapses.
so there are subsets of synapses in here that are always going to get the same value. You can run this evolutionary algorithm until the heat death of the universe, and it'll never be able to assign different weights to these different synapses. It's a small detail, and maybe it doesn't matter, but it's a prod to help you develop your intuition for how this works. So when I said this is tying Neat's hands, there are particular points in the space of all sets of synaptic weights that it can never visit. Maybe it doesn't actually matter, but I know these authors, they've got great attention to detail, so they didn't do this. They did something else. How do we untie the hands of HyperNEAT here? There's different ways.
I would like offset a layer by a certain value, maybe.
Maybe. The authors here created CPPNs with 6 input neurons and still 1 weight at the end. And the name of these 6 input neurons in the CPPN is a strong hint. How are they giving the CPPN information about a single synapse now?
They're giving the coordinate of the presynaptic and postsynaptic.
Exactly. They're giving if they want to paint this synapse, they give the XYZ coordinates of the presynaptic neuron and the XY and Z coordinates of the postsynaptic neurons. This is also nice because some of these hidden neurons have a self connection. By definition, the length of that synapse is 0. The length of that synapse is 0. It's kind of strange, but there you go. Okay, so deep breath. Here we go. NEAT is evolving populations of CPPNs. Each CPPN in the population, is painting a space into a 6-dimensional space, which results in a regular pattern of synaptic weights back in 3-dimensional space. My goodness, complicated. But we've gone through the rationale for why. Okay, got 5 minutes left. A lot of lead up to the results. So I'm going to show you 2 videos in a moment. The first one is going to show you the most fit neural network that was evolved using HyperNEAT, what we just saw. They're going to compare that gate that they got against a control. So this is another algorithm that is very similar to the one that I just described to you, but it's missing the magic sauce. It's missing the one thing that they put in that they think is very important, which is what they called fixed topology NEAT. So in this control algorithm, there's still NEAT. It's still evolving populations. It's still evolving CPPNs, but it's not going to allow. It's going to allow for mutation and crossover, but not changes in the cognitive architecture. They're kind of weakening the HyperNEAT. Okay, here we go. I'm forgetting which one this is. We'll do this double blind, meaning I don't know and you don't know. This is the most fit one from either HyperNEAT or from FTNEAT, the control. You're all experts on legged locomotion now. Which algorithm of the 2 wins? Or which one is this? This one wins.
I think
It's
it's
HyperNEAT. This is HyperNEAT. Now, of course, you're not seeing the actual pattern of synaptic weights, but you can see the result of it, You can imagine similar or identical synaptic weights flowing from sensors in this leg to motors in that leg, and the same sets of synaptic weights flowing from another leg, sensors in that leg, to the motors in that leg, and so on. They're synchronized. So you'll have to trust me. There is a regular spread of synaptic weights within the three-dimensional space of this neural network, which results in a regular behavioral pattern. So all that lead up to this, we're biasing evolution to regular things in the neural network geometrically that produce regular motions in time and space in the robot. It's not a very useful robot, but for most of the things we actually want robots to do here in the real world, regular patterns repeated over and over again are a good thing. We also sometimes want slight variations as we slip in that pattern, which again, HyperNEAT can do. Okay, we've got 3 minutes left. Let's look at something a little more familiar. Here's another fitness curve. So we've got evolutionary time on the horizontal axis here. Remember fixed topology NEAT here. This is NEAT. It's evolving CPPNs of fixed topology. It can't add and remove hidden neurons to the CPPN. So the CPPNs are restricted in the kinds of regular patterns they can produce, and you can see that they suffer as a result. After 1,000 generations of FT-NEAT, it's only able to get the quadruped to move 3 body lengths. HyperNEAT, 7 body lengths. Here's HyperNEAT and some of the regular patterns it produces, and here's FT-NEAT that has restricted CPPNs that can't grow very much. There's some regularity here, but not as much. Okay. Let's see. I want to end with this last slide here. Okay. We've got evolutionary time again here on the horizontal axis. I want to focus on HyperNEAT for a moment. So I want you to focus on the 3 solid lines. Forget the 3 dashed lines for a moment. I want you to focus specifically on the solid line with X's. This is reporting the fraction of individuals in the population at any given time. We're evolving populations of CPPNs. This is reporting the fraction of CPPNs in the population that had fitness. These are CPPNs that had fitness values intermediate between the fitness values of their 2 parents. These are all the hybrids in which NEAT cut the Mac and the PC together, glued those pieces together, and ended up with a new laptop that was about as good as the original Mac and the PC. Almost 60% of the individuals in the population were offspring that inherited parts of CPPN from parents and did as well as their parents. This is trying this back to our discussion about NEAT, showing that this all works in the sense of we're evolving blueprints that describe things that paint patterns into robot brains that result in regular motion. And along the way, this evolutionary algorithm is is very often bringing together the best of both worlds, combining useful genetic material from both parents. Okay, that was a lot this morning. This is the most complicated evolutionary algorithm you're going to see in this course. I appreciate your patience. You have a quiz due tonight. You're working on A8 or D3. Have a great week off, and we will see you in a week and a half. Thanks.